everyone, welcome to the course on medical biomaterials. We will continue on the topic of biofilm, and as I have been telling you that biofilm is formed on implants, devices, even if it is placed inside the human system for a few hours leading right up to few years, and this biofilm could lead to inflammation, infection and rejection of the material. Biofilm can form on any type of material, be it polymeric, be it metallic, be it ceramic, and it can be forming at any part of the body. It could be a blood contacting device, or it could be orthopedic implant, or it could be a urethral, or it could be a drug delivery system. So, we have been talking about biofilms and what are the various issues related to biofilms, what are the components of the biofilms, and what are the different techniques that have been tried out in research labs for eradicating these biofilms. Biofilm leads to a lot of problems such as the bacteria can slowly acquire antibiotic resistance, because some of the bacteria which are right down inside the biofilm do not get the same concentration of the antibiotics, or do not get exposed to the same concentration of antibiotics as that is present at the surface of the biofilm. The nutrient amount also varies, the concentration of nutrients at the top as against the concentration of the nutrient somewhere in the bottom of the biofilm. The oxygen concentration also varies. So, there could be genetic modifications in the microorganisms which are present inside because they are exposed to different environments. Okay. There could be altered in the growth pattern, so they become persister cells. Okay. So, there are many issues, there are many reasons because of which biofilm is formed. Okay. So, how do you eradicate? There are different approaches. One is to develop antibacterial and antibiotic coatings. You could have slow release systems, you could have slow release polymeric systems, you could have antibiotics immobilized on the surface or coated on the surface and so on. Silver is quite used because silver is an antibacterial in the form of nanoparticle, in the form of ions. Deuteron, organic, inorganic nanocomposites, which is known to have antimicrobial properties and also anti-adhesive properties. What is this anti-adhesive? Anti-adhesive means it's that the material, the surface need not have antibacterial or killing effect, but it prevents the attachment of the microorganisms because of their altered surface characteristic, like altered surface energy. Maybe it is more hydrophilic and so on. That is called anti-adhesive. Use of polyethylene glycol, modified chitosan, antibiotic loaded polymer coatings, deuteronic coatings, formulations of silver, zinc, etc. For example, on inorganic materials like alumina or hydroxyapatite, immobilizing enzymes, because some enzymes like protease or lipase or even papain have either esterase activity or amidase activity or combination of both. So, they may be able to kill coating with antibiotics or immobilizing antibiotics. All these techniques help in killing the bacteria. Another approach is developing hydrophilic surfaces. So, such surfaces may prevent attachment of hydrophobic organisms, so it becomes anti-adhesive. Okay. How do the organisms acquire hydrophobicity or hydrophilicity on their surface? That depends upon the type of proteins that are present on the surface of these bacteria. There could be a lot of hydrophobic proteins present, which will prevent the attachment of these bacteria on hydrophilic surfaces. Okay, because the initial attachment is generally non-bonded interactions like electrostatic, van der Waals, hydrophobic interactions and so on. So, developing such surfaces or modifying the topography of the surface, okay, creating indents, creating nanostructures. By doing this, you are preventing bacterial attachment, because the question that is being asked is, why does for example, the shells which are living all the time in water do not get biofouled or biofilm formation, or if you take sharks and fish, why there are no biofilms on them? That is because they have lot of nanostructures present on them, which prevents bacterial attachment, which prevents large organism settling down and so on actually. Okay. So, different approaches have been looked at, and I have been giving you examples in the previous class of some of these approaches. We will look at again a few more of these approaches. For example, polyesters, okay, or polyethylene terephthalate. This is a polymer, okay, this is a terephthalic polymer. This is quite a lot used in large diameter vascular grafts. Suppose I immobilized sulfobutane, okay, sulfobutane has a N plus charge, okay, exactly a deuteronic type of charge. So, we tested this, modified these polyesters using this type of deuteronic material. So, this picture shows the attachment of bacteria on pure polyesters. This is after modification. So, we can see there is a lot of prevention in the bacterial attachment. Okay. So, this shows this could be one technique for reducing the attachment of bacteria. Let us look at another example. Twin 80, these are surfactants, okay, these are non-ionic surfactants, that is neutral surfactants. You can modify using them on polyurethane surface. Polyurethanes are widely used in urinary stents because of its flexible nature. I did show some pictures of urethral stents long time back. So, when you put these amphiphilic molecules, the contact angle of the surface, which was 90 degrees, quite hydrophobic, comes down to almost 20 degrees, very hydrophilic. Okay. So, this amphiphilic molecule like twin 80 reduces hydrophobic surface to hydrophilic and hence prevents the attachment of the bacteria. These amphiphilics do not have antibacterial effect, that means they do not have killing effect, but they are able to prevent the attachment because they have reduced the contact angle from 90 to 20. That means the surface energy increases or the material has become more hydrophilic once you immobilize these surfaces with this particular surfactant, a non-ionic surfactant. That is another approach. Let us look at another thing. So, suppose you take polylactic acid and in the presence of PBS, phosphate buffer, PBS is phosphate buffer, you get a certain attachment of say E. coli and or Protease mirabilis, okay. About in 24 hours we get about 10 power 6 CFU per ml of E. coli and about 10 power 10 CFU of Protease mirabilis, okay. But then in the presence of blood plasma protein, when this PLA is coated with blood plasma protein, the E. coli attachment goes up dramatically. You can see a 10 power 4 fold increase, okay, in both E. coli and Protease mirabilis increase. 10 power 4 means almost 10,000 time increase in the bacterial attachment, both types of bacteria, E. coli as well as Protease. So, the blood plasma protein enhances bacterial attachment, okay. So, you have to be very careful when you are talking about having biomaterial in the presence of blood plasma. Blood plasma protein has been found not only in this polymeric system, but also on pre-coating of titanium with human serum or plasma increases adherence. So, whole human blood causes significant alteration in biofilm structure, number of culturable and viable cells, okay. Plasma is observed to be main regulator of the transcription of genes in biofilm formation, maturation and immune evasion. This is another reference, okay. And it has been found that LB broth blood plasma stimulates formation of biofilm of microorganisms like Staphylococcus aureus greater than Pseudomonas greater than E. coli. This is another reference. So, you see all these references tells that blood plasma protein enhances the biofilm formation of wide range of bacteria like Staph aureus, Pseudomonas, E. coli. So, both gram positive and gram negative bacteria are affected tremendously in the presence of blood plasma protein, and they enhance the bacterial adhesion, okay. So, environment has a very serious effect on the attachment of bacteria. So, we looked at different types of case studies where different approaches have been tested to see how the bacterial attachment could be reduced. Okay. Now, if you look at this entire biofilm formation, there are many things happening. You are having flow, you are having mass transfer, that means movement of a substrate, and then we are having bacterial attachment detachment. Now, these bacterial attachment detachment, they may be having longer characteristic time, whereas the mass transfer diffusion, mass transfer kind resistance or mass transfer coefficient may be happening fast. So, they may be having lower characteristic time. So, if you look at the biofilm growth, biofilm maturation, attachment of bacteria, detachment of bacteria, biomass growth, they all operate at different time frames. Okay. And this is a very interesting picture which I have developed based on these references. Okay. So, the mass transfer, reaction, substrate diffusion, convection, they are all happening very fast. They are happening around 0.1 to say 10 seconds. Okay. Whereas, if you look at the biomass growth, biomass decay, detachment of biomass, this is happening in terms of 1000 or 10,000 or 100,000 seconds. They are much slower. Okay. So, if you look at the biofilm part, the mass transfer part, the hydrodynamics part, the mass transfer, the hydrodynamics are much faster when compared to the biofilm formation, which is much slower. So, we have a situation where the transfer of material, the mixing is faster, whereas the attachment of bacteria, the detachment of bacteria, the biomass growth is much slower, almost by 1000 to 10,000 times. Okay. So, let us look at the characteristic times for each one of these processes. Okay. So, let us look at this mass transfer, the time, the diffusion time, characteristic time, diffusion time is delta square by capital D. D is the diffusion coefficient, this is the thickness. Now, if you look at the convection, this is D by U, the characteristic time, convection time, again this is thickness, U is the velocity. The viscosity, because of which happens because of the viscous dissipation and hydrodynamics, delta square divided by kinematic viscosity, this is the thickness. Okay. So, these are things related to mass transfer and related to hydrodynamics. Now, if you look at the biomass growth, that is reaction, substrate utilization, and then biomass decay, biomass detachment. So, the reaction happens because of presence of substrate, because of the biomass. Okay. That is the characteristic time, the reaction time. The growth is 1 by mu, mu is the monod term, Td is the biomass decay is 1 by Kd, Kd is the constant related to the biomass decay, and Tde, that is detachment, is 1 by Kde. So, all these terms contains reactions, decay terms, monod term and so on, which are much slower when compared to the mass transfer, diffusion and viscous hydrodynamics, okay, which are much faster. Okay. So, in a typical biofilm growth, we are having all these things happening, very fast processes, that is the mass transfer, hydrodynamics, very slow processes, which is the bacterial growth, bacterial detachment and bacterial decay. It is very important to know this scenario because although we are predominantly interested in the biomass growth, we need to understand when this mass transfer does not play a role in biofilm, and when the mass transfer or movement or convection plays a role in the biofilm growth. Okay. But then there is a vast difference in these times, which is of the order of almost 1000 seconds, I would say. Okay. This is a very useful slide to get a feel of the characteristic times. we have fast processes and slow things happening actually. So, if you are looking at biofilm growth, we can completely ignore, because we can say the mass transfer happens very fast, the movement of material from the bulk on the surface also happens very fast, and so on actually. Okay. So, biofilm structure we are talking in terms of days, whereas hydrodynamic, okay, that is less than 1 second. So, the time for growth is much larger than the time for diffusion. So, we generally ignore that, okay, because the diffusion times are much smaller. Okay. Now, there has been some attempts to model the bacterial biofilm. We are not going to spend much time in modeling these bacterial biofilms. Models could be quite complicated. We can have heterogeneous type of models, and but then at least we will look at what are the assumptions on which the models are based, and what are the terms the literature has considered while trying to model this, and is based on this particular reference. So, what are the assumptions? They have assumed biofilm consists of active biomass, inactive biomass, and water. Okay. So, active biomass, okay, which is not biodegradable, whereas inactive biomass is related to endogenous decay, and it is a fraction of this active biomass. Okay. What are the assumptions? Biomass is treated as a homogeneous continuum. So, you have to remember that although biomass is heterogeneous, you may have live cells, dead cells, proteins, and so on, it is considered as homogeneous. So, inactive biomass is related to the decay, and the fraction of the active biomass is inactive biomass, and that is not biodegradable. Nutrient diffusion to biofilm is from the solid substrate. So, in this particular example, they have assumed that the solid surface is giving the nutrient, that means from the bottom, rather than nutrient coming from the liquid side of it. Then you can assume a monod growth model for the biomass growth, right. You remember you must have done in long time back in bioprocess, where you have mu is equal to mu max divided by K plus S and so on actually. Then you have flat surface, so area of diffusion is invariant with time. So, there is no change in the area of diffusion, but in a real situation, as the biofilm grows, area that is exposed to the movement of the nutrient can change. As the biofilm becomes bigger and bigger, the area per volume can become smaller and smaller, and so on actually. Okay, so, we have different terms. We can have a mass balance of active biomass, rate of change of the active biomass, that is you may have a d by d time term, increment rate of active biomass due to cell growth minus inactivation rate of active biomass. So, some active biomass is becoming inactive, and the biomass is growing because of the cell growth. This is these two. On the left hand side we have a rate of change of the active biomass. Let us look at the mass balance for inactive biomass. Where does it come from? It comes from the active biomass. A fraction of it becomes inactive. Inactive biomass increases as the active biomass becomes inactive. So, a fraction becomes inactive. Okay, that is quite simple. Mass balance of nutrient in the biofilm. The rate of change of the nutrient in the biofilm, that is accumulation of nutrient, diffusion rate of the nutrient into the biofilm. So, some material is coming in, substrate is coming into the biofilm due to this diffusion, and consumption rate of the nutrient, because the biomass or the bacteria makes use of this nutrient. Okay. So, this is plus, this is minus, that is very obvious. So, that is the rate of change of the nutrient in the biofilm. Mass balance of the nutrient concentration on the solid substrate. So, this happens because there is a diffusion that is taking place, okay, from the solid surface. In this particular example, the nutrient is coming from the solid surface. So, you have to remember that. Okay. So, all these terms come into the picture. We have the mass balance of the active biomass, part of the active biomass gets converted into inactive biomass. Okay. So, this there is no decay of the inactive biomass, so that remains constant. Then you have the nutrient that is coming in due to diffusion from the solid surface, then nutrient gets consumed because of as the bacteria grows. So, the consumption rate we can put in a monod type of rate equation. So, the left hand side will be the accumulation of the nutrient in the biofilm as a function of time. Okay, then we have the nutrient concentration in the solid substrate, the rate of change of the nutrient concentration in the solid substrate due to diffusion. Basically, this is happening because of the diffusion at the interface. Okay. So, all these terms, so we can have differential equations, it can be a non-linear differential equations, because when you talk about monod equation you can have a non-linear monod term here. Okay. Then we can have fraction of the active biomass going into the inactive form, and then for the mass balance of nutrient in the solid we can put in something like a fixed law term here. So, all these equations can be put together and they can be solved. So, we can get a biofilm, that is the amount of live biomass change as a function of time, the amount of dead biomass or inactive biomass as a change as a function of time, the amount of nutrient concentration inside the ball of biofilm as a function of time. So, it is quite a simple model, because we consider it as a homogeneous type of situation. Okay. So, it can be modeled without much difficulty. What is the use of this type of model? We can see how the biofilm grows, how much nutrient is getting consumed, we can see what is the rate of conversion of active biomass into the inactive biomass, we can look at the accumulation of the inactive biomass. So, all these can be done. So, there are different approaches by which you can model the biofilm growth, biofilm maturation and nutrient consumption, but I just showed you superficially how one can go about it. This is the philosophy of it. Let us not go too much into the biofilm modeling, but this is how it is. Okay. So, as the biofilm grows, you are going to have very complex structures. You can have a very uniform flat biofilms, or you can have biofilms with a lot of interstices for the nutrients to flow. So, we can have very complex structures, that is possible. That depends upon the rate of nutrient diffusion, that depends upon the rate of oxygen diffusion, and that depends also on the rate of consumption of the nutrients by the bacteria and the rate of growth of the bacteria. Okay. So, there is a factor called G factor, which is defined like this: G factor is equal to delta square mu max x divided by ds. Okay. Delta is the thickness of the biofilm, mu max is your monod term maximum, x is the concentration of the bacteria in the biofilm, d is the diffusion coefficient, this is the concentration of the substrate, that is the growth rate limiting substrate in the bulk of the liquid. So, if we calculate this, so biofilm thickness, I know the mu max, that is the bacterial growth monod kinetic term, I know the concentration of bacteria in the biofilm, I know the concentration of the substrate in the liquid side of it, I know the diffusion coefficient. If G is less than 5, okay, if G is less than 5, then you will get dense solid biofilm matrix and a flat surface. Okay. So, if small g, if the G is small, that means this term is very large. Okay. So, the diffusion is diffusion of this substrate from the bulk on the surface of the biofilm is very fast. So, it is more of a reaction limited. For small g means this term is large, this term is small. So, this term relates to reaction, this term relates to the diffusion. So, small g means the diffusion term is much larger than the reaction term. That means the diffusion is not controlling, the reaction is controlling. So, we get very dense solid biofilm. Now, if the G is large, that means reaction is much larger than the diffusion. Okay. So, reaction is limited by the diffusion of the substrate from the bulk on the surface of the biofilm, that is when large G, then the biofilm will be complex structures such as mushroom clusters and channels. Okay. So, the growth is very fast, so the substrate is not enough for the bacteria to growth. So, the diffusion is the limiting factor, that means diffusion of the substrate from the bulk onto the surface of my biofilm, then you are going to end up with very complex structures of biofilms with mushrooms, clusters, channels and so on actually. Whereas, if the reaction is slow, that means diffusion is much faster, so you are going to have a very thick flat solid biofilm matrix. This is also a very useful term to have, this factor. It tells you which is controlling the biofilm processes. Is it the diffusion of my substrate, the limiting substrate which is helping the bacteria to grow, the diffusion of that from the bulk onto the surface of the biofilm, that is controlling, or is it the rate at which the bacteria grows, the biomass increases and the biofilm starts building up, which is controlling this or that. So, based on that we can also tell what type of biofilm structures you are going to have, whether you are going to have a very flat biofilm structure or whether you are going to have a mushroom or channel like biofilm structures. Okay. So, that completes what I wanted to talk about on biofilm. We spent quite substantial time on biofilm because, as I said, it is the most important topic, especially in the area of biomaterial implants and devices, and many materials fail in early days because of formation of infection, biofilm, development of persister cells which cannot be easily eradicated by simple antibiotics. The concentration of antibiotics required may have to go up by 10 times to kill the bacteria that is present and well developed inside the biofilm matrix, which contains exopolysaccharides and proteins and so on. So, we will talk about other topics in medical biomaterials in the next class. Thank you very much for your time.